Hi, my name is Lucas Kreiss, and today I want to tell you a bit about the identification of immune cells based on label-free multiphoton imaging and deep learning. And this is some research that has been done in collaboration with Duke University and the Friedrich Alexander University in Erlangen. As an introduction, let's quickly look at the conventional histology. What people are doing is they extract tissue samples, for example via endoscopy, put it into thick blocks of wax or some kind of medium, slice it into very thin sections that are then stained for this high image contrast, where you can look at individual cells and the entire tissue structure under normal optical microscopes. And then doctors would use those images to derive a diagnosis. What we did in our work in Germany is to use label-free multiphoton imaging for this procedure. So let's look at a cross-section of the colon mucosa. We can look at collagen fibers in the connective tissue using second harmonic generation, or we can use the natural autofluorescence from proteins like NADH or FAD. And then if we superimpose all channels, we see this nice three-dimensional image of the tissue structure, looking at glands, immune cells, and collagen fibers in the connective tissue. I said it's a three-dimensional technique, and this is a 3D rendering of the same acquisition, showing you that we can look actually a few hundred micron deep into the tissue. We did look at many different disease cases of this imaging technique. So let's first, as a comparison, look at the healthy control. Here we have many epithelial cells at the surface of the colon mucosa, basically closing the tissue and having a healthy barrier against the bacteria and pathogens. And if we look at the very severe inflammation, we can directly see the difference, right? There are almost no epithelial cells. There are a lot of immune cells and dead cell debris, and basically only the naked collagen matrix is still there. We can look at many different other types of inflammation, so for example, chronic inflammation or any other subtypes that the people use in clinical research, and we can always see that there's a distinct difference in this structure as compared to the healthy control. And as I said, it's a three-dimensional technique, so we could just zoom a bit deeper down into the tissue, and we see the same structural changes during these diseases. Yeah, so on a structural level, we already succeeded in this optical histology, basically. Let's compare the workflow. This is the conventional histology of actually staining and slicing the tissue. And this is our workflow where we just acquire the biopsy, we do the label-free imaging, and we directly get our 3D high contrast images. This led us to the question, how specific is our signal actually? Yeah, on a structural level, it works very well. We could see the differences. But can we actually make out differences in the autofluorescence based on the different type of cell that we have? And here we were working with a very talented student in our lab, and she isolated immune cells, looked at the 2-photon autofluorescence signature with the same technique that we used in the images, and we could show that we can actually differentiate some immune cell types. So I would generally conclude we can differentiate between innate and adaptive immune cells, whereas we can probably not really distinguish between the different subtypes of T cells. But there is evidence that we can differentiate different larger branches of immune cells. And this led me to continue this project and to look into something called virtual histological staining, or more broadly, digital staining. There is one great review by Oskin's group, and we also just recently published another review on this topic. Generally, what is done in this field is you perform any type of sample preparation and imaging, let's just call it the input image, and then you train a deep neural network to translate this input image into a specific target image. Again, the workflow, conventional histology with all the workflow specific to your actual staining that you do. Then our label-free optical microscopy, again specific to the optical contrast mechanism of our multiphoton imaging, for instance. And now digital staining would allow you to translate from any type of input domain, for instance label-free, to a more specific or some type of different target domain. This works usually in a supervised machine learning process, although some people also use unsupervised machine learning. Generally, what you do is you have these paired images of inputs and targets. So for instance, label-free and specific stain. You train your model, you validate it, you adjust the hyperparameters, all that stuff. And then when you deploy the model, you have a trained model that is able to predict the target images based on the input. So to show you some examples, here in this matrix, we have the input image contrast that is being used. So different label-free techniques, but also some stainings. And here are the target domains, which are mostly some specific immunohistochemical stainings. One example would be to use single photon fluorescence to predict H&E images, which is the one example here. Very commonly, people use phase contrast as an input to predict some kind of antibody immunofluorescence stainings. But it can also be used to do stain-to-stain transfer, for instance, to predict immunohistochemical stains based on H&E images. What I found in this literature review is that the field of nonlinear imaging, especially multi-photon imaging, to predict immunofluorescent stains is still underexplored. And this led me to start this research, which was recently funded by the European Union for the project called Multi-Photon Imaging with Computational Specificity, or MIX. And this name, of course, is a homage to PIX, a work from the late, great Popescu, Phase Imaging with Computational Specificity. I want to show you now the first preliminary results from this project. And for this, we started with a cell culture of two different cell types, neutrophils and T cells, where the T cells were specifically stained with an antibody marker. We then performed our standard label-free multiphoton imaging. We got these label-free images of NADH and FAD, but we also captured images of this T cell marker just based on a spectral separation of the fluorescence. These images were then cropped, so basically for each single cell I had one small image patch, and you can already see the differences. So in this case, this would be a marker positive cell, this would be a marker negative cells, and respectively you can also see the differences in the fluorescent and the natural fluorescence. I used these patches as my preliminary data set to train a model. It's relatively small, but still it has at least 5,000 cells, so it's not too bad. It's a bit unbalanced, there are more neutrophils than T cells, but it's also acceptable. I then used these patches for a patch classification. So basically performing conventional image processing and thresholding to derive the annotations for each patch. So just one single marker. So this patch altogether is a T cell, this cell altogether is a neutrophil. And then I fed the label-free images into a deep neural network with this annotation as the marker. When we look at the results, this was done in a 5-fold cross-validation loop. So we can see that the area under the ROC curve here for the validation case is already 0.89, so it's pretty good, considering that we have this preliminary set of very small data set size. If we look at the precision recurl, it's even a bit better with 0.95 area under the curve for the validation set. If we translate this into a confusion matrix, where we have the ground truth here, so the ground truth based on the actual biochemical marker, and the prediction based on the label-free input here, we can see that most of the cells were actually classified accurately, with some, of course, misclassifications in the training and the validation, but most of the cells were accurately predicted. So in this small set, we can already conclude, multiphoton imaging is able to distinguish between neutrophils and T cells in this preliminary test. What we can also see, of course, here in the confusion matrix is the cells were imbalanced in the data size, which I already told you. This brings me to the outlook. What do I want to do in the future with this project? Of course, we need more data. We currently have this limited data set of like 5,000 images of T cells and neutrophils. I want to get a lot more data from many different cell types, B cells, macrophages, dendritic cells, and so on. We need to get more data per cell type, but most importantly, we also need to get data from different imaging systems. Yeah, we want to test how general this model is able to predict immune cells, not just on one specific microscope, but on many different microscopes. And moreover, the long-term goal, the far vision of this project is to enable endomicroscopy with computational specificity. So I told you this digital staining works well, but what I really want to do is apply this technique to endomicroscopy. Already in 2019, we published this endomicroscope, which is just an endoscopic version of our label-free multi-photon imaging, which is enabling us to get these 3D label-free images of the living tissue. So even removing the need to acquire biopsies altogether. And if then we could apply some digital staining model like this to this type of data, this would really open up the possibilities for in vivo imaging at high specificity. I hope I could convince you that this is a very promising route to go, and yeah, keep up to date with the latest progress. I have to thank a lot of people who helped me with this research. There's one group in Erlangen from the medical department. There's another big group in the technical faculty for the imaging. And currently, I have a lot of support from the computational optics lab at Duke for the machine learning aspect. These are the people, the names to help me. And again, here's our project website. So please check that out to keep up to date. Thank you very much.